The last video talked about the pyruvate dehydrogenase to get us to acetyl-CoA. So we now have acetyl-CoA and we're ready to actually start getting into the TCA cycle. You'll notice that even though it is a cycle, since we're coming from glycolysis, we're going to go ahead and number these starting with citrate synthase, the first reaction being that which is going to add the acetyl-CoA that we get from pyruvate that we got from glycolysis. Citrate synthase is going to be our very first enzyme that we're going to start off with. Citrate synthase is classic CoA chemistry. We took a look before at how CoA will make our reaction more exergonic. In this case, the alpha carbon of the acetyl group in acetyl-CoA is going to be acidic and can be deprotonated to form that carbanion. And then that's going to be the strong electrophile that can attack the alpha carbon of oxaloacetate, which will yield us the citral-CoA. That will then go ahead and hydrolyze and get rid of the CoA to leave us with citrate. Again, this is going to have a large negative delta G value and is going to be a site of regulation. NADH and succinyl-CoA will be allosteric inhibitors. We'll see both NADH and succinyl-CoA later on in the cycle. So as you might imagine, NADH will be an inhibitor because part of what we'll get out of the TCA cycle will be a lot of NADH. So anytime that we have enough NADH in the cell, the TCA cycle will be inhibited. Similarly, succinyl-CoA is another thing farther down the line in the TCA cycle, and so you'll have that negative feedback loop. The second reaction is that of Aconitase. This is going to take us from citrate to isocitrate. It seems like such a small move, but citrate's a really poor substrate for oxidation. So since part of the goal of the TCA cycle is to generate NADH, we need to have catabolic oxidation. And citrate's simply not very good at that. Aconitase is going to isomerize the citrate to yield isocitrate, which will then be more easily oxidized. Aconitase also uses an iron-sulfur cluster. These I do think are really cool. So iron-sulfur clusters, this one's actually going to help participate in acid-base chemistry. So the iron-sulfur cluster is bound to the protein by side chains of 3 cysteine residues. Binding to the iron, the vacant position that's in the cluster is going to activate the aconitase. So 3 of the irons are bound with cysteine, leaving you a 4th iron. And that 4th iron is what's going to actually work on the substrate itself. That iron is going to coordinate the C3 carbon as well as those hydroxyl groups and act as a Lewis acid and accepting an electron pair from the hydroxyl group to make it a better leaving group. Still on aconitase, fluoroacetate is actually an extremely poisonous agent. It's a rat poison and it blocks the TCA cycle. But it's interesting because it actually gets in kind of early and can be accepted to acetyl-CoA, and then citrate synthase works on it just fine. So it's a poison, but it doesn't actually act immediately. Once it's converted to fluorocitrate's a potent inhibitor of aconitase. So this will go ahead and inactivate aconitase, but it'll go through acetyl-CoA synthetase just fine, and it'll go through citrate synthase just fine. But because it will essentially inhibit and stop aconitase, it'll actually form a covalent bond in there. It's known as suicide substrate, or even in this case, even a Trojan horse inhibitor, or again, that irreversible inhibitor. So again, multiple names to say that this is going to form a covalent bond in the active site of our aconitase, and our aconitase enzyme, if it interacts with that fluorocitrate, won't work anymore. The third reaction. So we have our isocitrate. Isocitrate is then going to go to alpha-ketoglutarate. This is the first oxidative decarboxylation in the cycle. This is fairly classic NAD+ chemistry. So we are removing a hydride from our isocitrate to get to, in this case, oxalosuccinate, which will then be decarboxylated to alpha-ketoglutarate. So just note here that it is the oxidation that actually happens first. And then the beta decarboxylation reaction releases the central carbon as that CO2. So do pay attention here which carbons are getting released and what our order of operations are here. This enzyme can also be inhibited in a feedback loop from NADH. It's also inhibited by ATP, but activated by ADP. Reaction 4, we're going to take alpha-ketoglutarate to make succinyl-CoA. So this is alpha-ketoglutarate dehydrogenase. This is going to catalyze the second oxidative decarboxylation of the TCA cycle. This enzyme is nearly identical to pyruvate dehydrogenase, both structurally and mechanistically. So if you'll notice, we again have 5 different coenzymes. Thiamine pyrophosphate, coenzyme A, lipoic acid, FAD, NAD+. So we have those same 5 coenzymes, and it's also having a dehydrogenase reaction. It's the same thing. So this actually has the exact same mechanism as pyruvate dehydrogenase. The number of subunits is a little bit different in here, but you still have that notion that you have a complex with lots of different parts of the 3 different enzymes that make up the alpha-ketoglutarate dehydrogenase complex. So again, you still have an alpha-ketoglutarate dehydrogenase, where before we had a pyruvate dehydrogenase. Now we have a dihydrolypyl transsuccinylase, as opposed to that transacetylase. And now there still is the dihydrolypyl dehydrogenase, because that last E3 portion of this complex is still going to be to reoxidize the lipoic acid that's in the E2 enzyme. Reaction 5, Succinyl-CoA Synthetase. Once we have our Succinyl-CoA, what we're going to do is have a substrate level phosphorylation to go from GDP to GTP, which will also release CoA. So that's going to leave us with succinate. Our Succinyl-CoA is a high energy intermediate. That's why we can actually make that nucleoside triphosphate during this reaction. This is going to be the only time that we have a substrate level phosphorylation in the TCA cycle. This GTP can actually then be used to phosphorylate a DP to make ATP by a nucleoside diphosphate kinase. In plants and bacteria, you actually just, in this reaction, you actually usually just use ADP to get phosphorylated to ATP directly. So whenever we talk about the TCA cycle, sometimes you'll see them say that you make a GTP, sometimes you'll hear them say that you make an ATP, sometimes they'll just be vague and say a nucleoside triphosphate. As far as I'm concerned, we're going to basically use that GTP, ATP interchangeably here, so it doesn't matter to me which one you're talking about, because essentially they're interchangeable since you can go from GTP to ATP. Since this reaction does involve that substrate level phosphorylation, we're going to need an inorganic phosphate. So what happens here is the phosphate goes ahead and nucleophilically attacks the carbonyl. That actually kicks out the CoA. From there, a histidine then is going to nucleophilically attack the phosphorus group of the phosphate, and that'll go ahead and release succinate. You're then left with a phosphorylated histidine residue. So then in order to get our histidine residue back so the enzyme can repeat this process, we have GDP going to GTP. And that's what then releases the phosphate group off of the histidine.